this lecture. We are excited to have a speaker today from Netherlands, Aldona Mzyk. Interestingly, Ramana Shiragl, with whom Aldona worked together, gave a lecture 1.5 years ago on using fluorescent nanodiamonds for detection of free radicals in live matter. So I realized today suddenly that it's 1.5 years past, and time flies so quickly. And today we have another lecture with completely different topic from the same group. And this is very exciting that people don't stop their progress, that they continue to generate results, that because of pandemic, we did not stop our development. So this is a great thing happen. So I would like to start with introduction of Aldona. So Dr. Aldona Mzyk is an early career researcher with a background in biotechnology and material science. She enjoys working on fundamental scientific questions, but the real fun for her starts when it comes to bringing science into industrial and clinical reality. Over her years in academia, she has become focused on the real-world outcomes of her work. When she started her romance with science, the idea was all about how it works. However, there is limited amount of funding for how it works. So she had to switch towards make it work. This pragmatic approach has led to the award of 2 Polish through 3-year scholarships to pursue her PhD thesis. In this time, she developed biomimetic coatings, which is the subject of her ongoing collaboration with the Foundation for Cardiac Surgery Development on functionalization of the Polish Religa heart assistant devices. After her PhD, she was hired as an assistant professor in the Institute of Metallurgy and Material Science, Polish Academy of Science in Krakow, where she has been working on biomimetic materials and surface engineering methods to be applied in cardiovascular implants and devices. She was lucky to be awarded with a personal grant from the Polish National Science Center to pursue fundamental research on cardiac progenitor cells response to biomaterials. The biomaterial part of her project found interest of the 3D printing company Atmat in Poland. The team secured funding from the Polish National Center for Research and Development, which gives her the opportunity to work part-time for the company at present time. In September 2019, Aldona joined Professor Ramana Shiragl group in the Biomedical Engineering Department at the University Medical Center Groningen, the Netherlands, to work on fundamental topics with imperfect nanodiamonds. However, her drive to push forward tangible applications persists, emphasizing directions that have clinical relevance, which will be the topic of her talk today. Aldona, please, the stage is yours.
Thank you, Olga. So thank you for that introduction. And of course, for giving me the opportunity to share with you with that broad audience from around the world, my work and work of my colleagues. So welcome everyone. Today I'm going to talk about how we can use the fluorescent nanodiamonds to detect free radicals, and I would like to try to show you, convince you that the free radicals could be a great biomarker for drug screening and a clinical diagnostic. So as I know that many of you are like diamond enthusiasts for over years and with a huge experience and knowledge, but maybe few of you are just join us and will become the diamond enthusiast soon. So I decided to explain shortly how we can detect free radicals with nanodiamonds. So as that's also the core part of what our group is focusing on. So I will start with defining what are the free radicals. So those are kind of the active molecules that possess at least 1 ampere electron. And those one, because they possess this 1 ampere electron, that causes the magnetic noise. And this magnetic noise, so-called, could be detected by fluorescent nanodiamonds, thanks to the fact that they have kind of special defects in the structure called the color centers. And we are working with the diamonds that have nitrogen vacancy centers as a defect in the crystalline structure. So nanodiamonds, so those vacancy centers, nitrogen vacancy centers, provide the diamonds with this very special optical and magnetic properties at the same time, so they can be used as a probe to detect free radicals and magnetic noise. So how it's possible? So when we shine the green laser light on a nanodiamonds, they emit the red photons, but in the same time, we polarize the electron spins. And as we're using the sequence of the green laser pulses, and between this pulses, we just wait a little bit, right? We wait. For what we were waiting, we'll wait that the polarized electron spins, they repolarize, they go back to the previous status. And then we are interested to say how long it takes those electrons from repolarize from polarized to go back to from polarized to repolarized state. So this is what we measure, and this time will be different. In the presence of the free radicals, depends on how much of the free radicals we have in the surrounding of the fluorescent nanodiamond probe. If we have higher concentration of free radicals around the fluorescent nanodiamond probe, then this relaxation time will be shorter. And this concept is a kind of analogy to the what we measure in T1 in MRI method. But for our method, so nanodiamond magnetometry, to use nanodiamonds, we don't need this big, huge equipment that the people that specialize in MRI they're using. But we need a little bit different setups. And I heard a few times from some people like, oh yeah, you know, this method is like so fancy, high tech. So why you want to bring it to the biomedical field? Why you want to do that applied in the clinics or drug screening? It's like really high tech. It's like quantum sensing method, right? I say, yeah, okay, maybe it's a very smart, fancy science behind, but the method is pretty simple. I mean, I learned it. I believe many people can learn it, how to perform measurements with an understanding and how to analyze the data and how to derive the conclusions from it. So what I'm trying to say is, of course, we need to deliver somewhere, target our diamonds near the source of the free radicals, as the probe needs to be near the source of the free radicals to be able to detect. And we need to provide the source of then green laser light. And how we provide it, we just basically build the confocal microscopes. So this is why, as in general, in domain of life science, we're using the confocal microscopes every day. And this is why I'm saying this is not that high-tech fancy. So it could be, that could be used in a clinical or big pharma scenario in the future. That is at least my opinion. So I also started to talk like that's possible to use this technique in a different scenario, not only in lab in the future, but why do we need it? Do we need it? Do we need nanodiamond magnetometry to detect the free radicals? Yes, we need. Why we need? Because free radicals in living cells, they play a big role there. They are involved in development of many types of diseases, but they are also kind of the molecules taking the part in the physiological processes. So they are signaling molecules. But they are very difficult to detect. They are short-lived and very reactive, as I mentioned before. And there are at the moment no really specific probes available to detect the free radicals in living cells. So there are some probes that we can use to detect the reactive oxygen species or reactive nitrogen species, but not the free radicals per se. So, and of course, those have many different disadvantages. So with the current techniques, the most important thing is that with current technique, we cannot just measure radicals in real time, and that we can do with the nanomagnetometry. So basically, I hope now that I convinced you already a little bit that this nanodiamond magnetometry potentially in future could be used in a broader, so it could be used by many different companies or in a clinical scenario. And it's kind of providing a new quality in our detection methods. So for now, I would like to share with you some examples of our work to back up what I just said, that's possible and we are on a way there. We're trying to bring the methods, so nanodiamond magnetometry, to detect the free radicals in living cells to measure the free radicals in a clinical samples or to screen the drugs efficacy. So one of my first example will concern our research on infertility. Why we are interested in studying infertility with nanodiamonds? Because that's a kind of big problem around the world. It's like 1 in 6 couples had some issues with an infertility, so they can't just conceive easily. And that's cost a lot of money, the treatment for couples. It's like in Europe, it's average per treatment, for example, for IVF with some medications, 20,000 euro. And it's a case in important thing is like that in 4 in 10 cases of infertility are attributed to men subfertility. So it's not, so 50% of the cases is because of something's not working on the male side. And among all these cases, 30% are related to increased elevated level of the free radicals. So there is the link between the free radicals level and men subfertility, infertility. So, how the free radicals contribute, or where they come from, and how they contribute to male subinfertility. So there are different sources. Of course, there are some internal sources of the free radicals, so our immune system, so the man immune system produce them, so leukocytes, and also some immature spermatozoa, they produce quite a lot of free radicals. We provide them by just enjoying our life, smoking, drinking alcohol, or by being exposed to different environment factors, some toxins, radiation, for example. And those free radicals are not only the bad guys in that story. So they are, perhaps you all heard that the free radicals cause the lipid peroxidation of spermatozoa. They can cause DNA damage or even apoptosis, which is the death of the sperm cells. And that leads to male infertility, but they also play a very crucial physiological role. So some level of free radicals is there. They are produced by functional spermatozoa, by healthy spermatozoa, and they are involved in capacitation, which I will talk later on a bit. So it's kind of maturation of sperm cells, in acrosome reaction, in motility of the spermatozoa, and also the binding of sperm cells to the oocyte and the fusion of those two cells. So those are important also in that processes. If we look deeper at the cellular level, if you look at the sperm cell, there are 2 different main sources of free radicals. So free radicals are produced, of course, by mitochondria that are located in a midpiece of the sperm cell, but they are also generated by NOx5 machinery, which is located in a membrane in a head region of the sperm cells. So now the question is, still, I mean, there are many questions in the field of reproductive biology, of male reproductive biology, related to the what's the role of the free radicals, how they work, what kind of free radicals are important for what type of physiological process. And in our research, we wanted to first answer at least one, which is very important, which is what's the role or what's the source of free radicals involved in a sperm capacitation. That those free radicals comes from NOx5 that I mentioned is located in the head of spermatozoa cell, or it's the ROS free radicals comes from a mitochondria in a meat piece. So we wanted to prove that the magnetometry works to detect the free radicals at a certain level that produced during the physiological processes, and at the same time also answer the fundamental scientific questions like what's the source of the ROS that is involved in capacitation. So first place, we work with the animal material, so with the boar sperm cells. Now we have the ethical approval, we just got it. So we will start our clinical trials, but I will show you some research that was done on boar sperm cells and will be translated now to human situation. So before we start measurements like the relaxation time, so the changes in the free radical levels, we had to answer also like check if we apply nanodiamond probes we treat the sperm cells with them, if they will be still alive, if they'll survive, or if that's something we change in them. So basically we wanted to screen by like perform some biocompatibility screening experiments. So this is what we did. We treat the sperm cells in different stages, so uncapacitated, and after we trigger in vitro capacitation process with the nanodiamonds. We test different kind of sizes of fluorescent nanodiamonds from 40, 70, 120 with different surface terminations. But here I will mainly focus on showing the results of biocompatibility and then further experiments with 70 nanometer fluorescent nanodiamonds with two different type of surface modification or the surface termination. So oxygen terminated and amino terminated FNDs. So we perform metabolic activity assay, and then for two different concentration, two different surface terminations, and for one type of the size of the nanodiamonds, and then we found out there is no adverse effect of those diamonds on the sperm cells. Then we also check if what happened with the membrane. Maybe those diamonds attach. That's what we wanted. They attach to the membrane of sperm cells, or something else happened. But if if then the cell stays alive, is the membrane integrity fine? If this membrane still integral? And we found out that those variants doesn't cause also any adverse effect in membrane integrity. So that was a great encouragement to continue our research. We wanted to know where the diamonds attach. So we took some confocal imaging. So we use the property of the nanodiamonds as they are fluorescent. So you can see here them as these red dots. So we apply here 70 nanometer nanodiamonds of two different type of surface termination, with two different type of surface termination, oxygen or amino terminated nanodiamonds. And then we found out that in any case, for both cases, the diamonds preferentially are located attached to the head, in the head region of the spermatozoa. There is also some difference between how many diamonds attached to the spermatozoa depends on the surface termination, and if they form aggregates or not. So for amino terminated, you see there are smaller diamonds, they don't aggregate that easily, and there are more at the surface. Also, we found out that the nanodiamonds attach preferentially to, there is preference of nanodiamonds to attach somehow higher affinity to the surface of the spermatozoa that have kind of characteristic structure meat piece with very well visible mitochondria helical spiral, let's say, not to the one that the meat piece is quite smooth. So that was also quite interesting that we found something like that, that the diamonds just don't attach to any type or all types of the spermatozoa that we have in the population. So finally, we knew that the nanodiamonds it's fine to use the nanodiamonds, that they are located in the head of the spermatozoa. We said, okay, great, because we also want to detect we want to differentiate if the free radicals comes from, what is the source of the free radicals. So that's good enough to continue with the measurements, and we perform some T1 relaxometry measurements with, again, both type of diamonds first. And yeah, and we use as a control the spermatozoa sperm cells in incapacitated stage. So they're not triggered to produce that much free radicals. And then we also trigger them to produce more free radicals. And we want to prove if we are sensitive enough to detect that changes in the free radicals concentration.
Aldona, may I ask a question? Maybe you can elaborate a little bit more on these capacitated and incapacitated states, because we are physicists, so it would be great to know a little bit more of biological background.
Okay, so I'm sorry for that. Yeah, my bad. I think I try to say like the capacitation means like it's kind of maturation. So it's very difficult to define what that exactly is, but it's related to changes in the membrane of the sperm cells. So there is the change in the fluidity of membrane, and membranes are organized. And that's an important step, that the sperm cells is then able to get to the oocyte and bind to it and fuse with the oocyte. So it's important for fertilization, basically. So capacitation is like kind of when sperm is capacitated, means it's like a more mature, more ready to fertilize, to fuse with oocyte. And incapacitated is like kind of standby status. So yeah, so I hope now it'll be more clear, and sorry for that again. So yeah, so we had this incapacitated state, and then we trigger capacitation by treating the sperm cells with the capacitating medium. This is also part of kind of standard procedure that is used for IVF. So we trigger that capacitation, or that happened in oviduct. So we triggered the capacitation and we measured T1 over time, and we found out there is a drop in the T1 values over time. So that corresponds to increase in the free radicals concentration over time. So basically that way we prove that we are able to measure this physiological generation or changes in the generation of the free radicals by spermatozoa during the capacitation process with the fluorescent nanodiamonds. And then here we decide, like, both type of nanodiamonds with different type of surface termination worked well, but we found out that they're here in case of amino-terminated diamonds, somehow we got a more, let's say, noisy result. So we prefer then continue with just the oxygen terminated FNTs. So we brought that FNV, we brought that knowledge further, we move on. And here we wanted to prove, okay, if we can turn on, like, really, because that was not proving what's the source of free radicals that they take part in the capacitation or are responsible for this capacitation. So if that comes from NOx or that comes from mitochondria, we still didn't know, of course. We just proved that the method will work. So to check that out, then we decide to inhibit or activate both of sources separately, so NOx5 and mitochondria, and we use some chemical reagents that are capable to do that. So for NOx5 regulation, we inhibit NOx5 with treating sperms with apocynin, or we activate it by treating the sperm cells with progesterone. And for mitochondrial regulation, we use for inhibition, we use CCCP reagent, and for activation, diazoxide. So basically, here is the scheme of with some numbers. So number 1 is pointing out the NOx5 regulation potential mechanism with apocynin, and then below with the progesterone. So that's basically more or less how it works, presented here. And this is on the right side. You will see here how we regulate mitochondria. So it's just like for some people who like to have a brief look like how mechanism works. But I'm not gonna go really into details, maybe in discussion if someone's more curious about it. But I just wanted to share with you the results of the T1 measurement, because I think that's the most interesting part then. So we perform experiment as the sequence of measurements. So we had our spermatozoa, nanodiamonds attached, located in a head region, and then spermatozoa were incubated in this medium that promote them to stay like the standby medium, so incapacitated state for some time. And then we measure. So that was our initial measurement. Then we add to that environment, to that situation, we add the inhibitor or activator of NOx5 or mitochondria, and then we kept measuring. And then after time, using the same diamond, and then after time, we trigger capacitation process, so maturation. So we expected increase of the free radicals. But you need to remember that we are located with our probe in the head region. So I would say perhaps we measure from NOx5, right? Not from mitochondria. Only if free radicals from mitochondria are just able to diffuse or migrate close enough to our probe. So this is what we got. Those are the results of regulation of NOx5 and measurements of the T1 and mitochondria below for inhibitors and for activators. And based on that, I can say that we found out what's the main source. So we proved what's the main source of the free radicals that are involved in capacitation. But before I will say that, was that the source, perhaps you already see it, I also have to say that we are very surprised by the behavior of the progesterone. So that's something unexpected. We found out that by using the progesterone here, we didn't expect, when we had the incapacitated status of spermatozoa, we thought like, oh, progesterone not gonna work. Progesterone needs triggers for capacitating medium, which is a calcium. So it needs external source of calcium to work, to trigger capacitation further. But we saw it without external source of the calcium. So that also gave us kind of extra discovery, like that was an extra discovery we didn't expect, that the progesterone basically works and perhaps opening, that's our hypothesis, that is opening cut sperm channels. This is what basically progesterone is doing, but it's opening these channels in a containers, in intracellular containers, that are sources of the internal sources of the calcium. So it's not that progesterone only works with the calcium that is provided from environment, but perhaps it also works for calcium, which is stored in these vesicles inside the cells. And I think that's a quite interesting thing we found out and were to explore further. But going back to the main conclusion of that experiments using nanodiamond magnetometry, we found out that the NOx5 is the main source of free radicals capacitation, as we didn't really register significant signal when we inhibit or trigger mitochondria. So basically, schematically saying that, like presenting from this scheme, you can see like, yeah, this is what we got from the T1 measurements. When we measure, we regulate the NOx5, and then we saw some signal, the T1 or changes in the T1 relaxation time. When we regulate mitochondria, there was no significant difference in the signal. Okay, so before I will go to the next topic, to give another example of how we applying the fluorescent nanodiamonds to free radicals and how that might have consequences for a clinical diagnostic and drug screening, I would like to sum up that with already I mentioned that was done on boar sperms, but now as we have the clinical approval and the concept for experiment, we'll really have a patient, the human material, and we measure and we will try to correlate what's the free radicals, if radicals have impact on male infertility, and if those free radicals, somehow the levels of free radicals produced by sperm cells are regulated by lifestyle factors, medication, and so on. So this is basically what we want to try to do, correlate a lot of factors, and one of those factors, one of those biomarkers will be, they'll be free radicals. Another example of our research activities is focused on detection of free radicals in a knee. So why do we bother to detect the free radicals in a knee? Because the free radicals are involved in a joint degeneration. So that's an already known fact that both rheumatoid arthritis and osteoarthritis somehow are triggered or progressing due to increased level of free radicals. There are still mechanisms how exactly that works are not really known, or at least some part of the stories. So this is why we are interested to go there, and especially to develop the future therapies. So to discover, screen the drugs, if the drug A or B works better for osteoarthritis or rheumatoid arthritis. So that part of my talk will be dedicated to the arthritis experiments with like more on that's on a drug screening side. So first place, we perform some control experiments to again try if the magnetometry will, we challenge the technique and if we can really use it for drug screening. So we need the good controls. So if the drug itself causes some redox processes or is just has a form of radical and will affect the measurements. So it was indeed the case. So I think that's quite interesting that maybe magnetometry not gonna be possible to use for all type of drug screening. So as we try two non-steroidal anti-inflammatory drugs, Ibuprofen, that all of you is well known to all of you perhaps, and piroxicam. And those drugs are in use for the arthritis patients are using them. In case of ibuprofen, we had some kind of change in the T1. We had a change in the T1 value during the measurements. So we quit on an idea to use it at some point. I mean, that's very interesting to know why. And then we did some more literature research, and we figure out that ibuprofen also can have this free radical forms, and there perhaps could cause this magnetic noise. But we continue with piroxicam for our clinical trials. So in this experiment, we obtained synovial fluid from the patients from UMCG. And those were patients with osteoarthritis and rheumatoid arthritis. And this, we perform again, like kind of sequence of measurements, of T1 measurements. So first place, we put the diamonds on a dish, and we measure what's the relaxation time of those diamonds in a dry state. Then we add synovial fluid from the patient, and then we again measure that we're using the same diamond relaxation time. And then we treat the synovial fluid with the drug, with the piroxicam, and then we measure again T1 over time. And this is what we got for osteoarthritis patients. So of course, you see here that after we add the synovial fluid, there is a response. So there is a drop in a T1 value that corresponds to increase in free radicals concentration, we think. And then after treatment with the drug, the T1 value, let's say, kind of go back to the initial stage. So basically we have less free radical after time. So it's kind of neutralization of the effect of generation of free radicals. We perform to compare nanodiamond magnetometry to more, let's say, standard techniques. So we performed DCFDA assay, which is a fluorescent assay, and we had the same trends. So in that case, DCFDA didn't work that bad. But you need to remember that this is basically a method that's used to detect the reactive oxygen species. It's the fluorescent method, so it's a lot. We deal with the bleaching, we deal with phototoxicity, and of course we cannot measure in real time or over time. So it has a lot of limitations, but proof that magnetometry works also. And then we had also patient with rheumatoid arthritis. So this synovial fluid also, when we detect a certain level of the free radicals, so drop in a T1 value when we treat the diamonds with the synovial fluid, or we add synovial fluid to the diamonds. And then we treat the synovial fluid with piroxicam, so with the drug. And here we didn't see any significant difference. So it's not like in osteoarthritis synovial fluid from osteoarthritis patients, there is no difference here. And that was similar for what we detect with the CFDA. Moreover, what we did as free radicals then in synovial fluid needs to come from somewhere. They come from, potentially they come from different type of cells that are in the joints. So chondrocytes, synovicites, and osteocytes. And in synovial fluid, we have those cells. So this is basically the source of our ROS that we detected right away directly in the synovial fluid, but also we culture. So we derive that cells from the synovial fluids, we culture those cells, and we perform similar type of measurements. So we treat the cells with in a culture after 1 and 7 days with the nanodiamonds. They taken up nanodiamonds, we let them a bit rest, and then we measure, we took a first measurement. We always use the same diamonds. So then after time, after a certain number of measurements, we're using the same diamond, we add the drug, and then we perform again the measurements after drug treatment. And then what you see here is the case for osteoarthritis compared to rheumatoid arthritis patients' results. And after 1 day of cell culturing, and in this case, we see that the drug works similar to what we observe for just when we detect changes in the free radicals level in osteoarthritis in synovial fluid. And the same situation was, same trend was for osteoarthritis patient. I mean, osteoarthritis patient cells versus synovial fluid. But as you can see also here, that remains that that here this change after drug treatment is much bigger and more significant than here. So rheumatoid arthritis cells from rheumatoid arthritis patients respond less to the treatment with the piroxicam. That situation was not visible anymore after 7 days of culture. So yeah, I mean, I believe that's because we just culture cells in vitro. So perhaps if there were some external factors in synovial fluid, they're not working anymore for sure here. So we somehow cut off potentially other environmental factors and other crosstalks interaction with other types of cells in the joints. So we don't see this effect here anymore. But that might be also, of course, something different. That's, I believe, worth to explore further. And as we don't see these differences, and it's very difficult to screen the drugs on the models of just the simple monolayers or single cells in vitro or in a cultures. And everyone's, of course, is moving a little bit forward towards the screening drugs and doing stuff or organoids. That's also our idea to do so. And we have one project or few projects running now on organoids. And in that project, will just show shortly what we started doing there, as we want to generate also cartilage organoids, and then hopefully maybe that organoids going to be derived from the patient cells, and in combination with the synovial fluid. So that will be perfect situation to combine like our small project outcomes that we already running, or we've been running in one, and move forward. And this is why we started that one. So far we have the iPS cells, so induced pluripotent stem cells. So these cells they were some type of the cells, they were differentiated, and then now we can differentiate them towards many different types of the cells we want. And in this case, we want to differentiate them towards the chondrocytes mainly. So we start the differentiation process, and as differentiation process, free radicals levels should change, but there's also a lot of questions remaining to answer here. How free radicals are responsible, involved in that process of differentiation. So this is what we start studying. Differentiation from iPS cells towards chondrocytes, towards cartilage organoids. And here I just have one slide, summary slides on that research part. So you can see that process of differentiation towards the cartilage organoid takes some time. It's 21 days, basically, and starts from pluripotent stem cells. It goes through definitive endoderm cells, spheroids formation, and then finally cartilage organoids. So I'm showing here only, I'm not sure if you see that well, but there are some diamond cells. I'm showing just the two first steps here because we still have a lot of images to process and take from that experiments, but we were able to monitor changes in the free radicals concentration, or rather T1 values. This is what you see here during this differentiation process. So you see first there is a certain level of the free radicals in that kind of cells, so induced pluripotent stem cells when we treated with the nanodiamonds, and then when we start differentiation process after 3 days, we observe drop in the T1 value that corresponds to increase in the free radicals concentration. That level of free radicals stays over 14 days, and then we observe that that's again the free radicals concentration is going back to slowly initial state. So cells has less free radicals, so maybe they are less metabolically active, or they need less free radicals signals in a differentiation process. So this is something we want to explore, and we are exploring further. That was done for nanodiamonds that were probably they were in some kind of transporting vesicles, endosomes or redoxosomes. But now we would like to also target nanodiamonds to mitochondria and check how metabolic activity during differentiation process change when do we, that's what we wanted to investigate, how the metabolic activity and production of the free radicals by mitochondria change over time during differentiation. Okay, that's all I prepare for today for you. I hope that you see that we are really step farther and closer to the drug screening concept and clinical diagnostics with the fluorescent nanodiamond and nanodiamond magnetometry. And at that point, I would like to thank my great mentor, Romana Shirhal, and all group members, because without them, that work couldn't be possible. And of course, funding organization for the same reason, and you for your kind attention. And I hope I will get and be able to take and answer many questions from you. Thank you for your kind attention.
Aldona, thank you very much, and congratulations on great achievements in this translational work, working with on real medical problems. And obviously, there is a very big advancement over last year and a half from what we heard a while ago, and how serious this Groningen group is to help to solve problems in infertility or in osteoarthritis So it's very impressive, really. So congratulations. I
Thank
would
you.
like to open our Q&A session. Please ask your questions. You can just ask straightforward, unmute yourself and ask the question.
Aldona, I have a question. Sorry, I'm on the wrong account, so it's Nicholas Nunn. But on your, I'm just trying to understand, when you labeled these spermatozoa cells, okay, do I understand that you're doing measurements on a single particle on a single cell?
Yeah.
And you're so how, like, I guess, how do you pull that off? Are these cells moving around, right, while you're trying to do that? So you're tracking an individual particle on a cell that's moving simultaneously?
Yeah, so yeah, okay, so yeah, it's a little bit of more technical part, but of course it's then you can imagine that if there are cells moving or something moving around, a particle moving, that might be problematic for measurement, so on. And so I understand where I think where your thought's going. So so no, it's like indeed single cell level. So we do perform experiments single cell level, single particles. And those particles are like kind of attached to the membrane, and cells are attached to so we immobilize the spermatozoa. So it's reversible immobilization. So it doesn't hurt them, let's say. But yeah, what we have to immobilize them. So because we, of course, tracking the particle, tracking the spermatozoa at the same time would bring a lot of, measurement errors, I would say. Yeah.
I have a quick question from the United States. You have presented several different clinical conditions, and the question is kind of not at all being an expert in this area, so trying to learn. When you talk about free radicals, are you assuming that the type of free radicals are exactly the same in each of the medical conditions? And when you test different drugs, they all address the same kind of oxygen radicals. There are many of those. They have different activity, reactivity, and they have very different short half-life time. So I wonder just if free radical is just a certain consistent groups of chemicals that are the same in all medical conditions.
No, of course you're right. That's a very good point. Thanks for bringing that here. Yeah, it's not. It's like for now, indeed, it's just very general statement that we measure free radicals. This is what we measure, but there are many. There are different types. They, as you said, different lifetime, and yeah, they are produced by. Yeah, they are just different types of free radicals responsible for different signals. So in medical conditions, there might be some, like for example, superoxide might be overproduced, or nitric oxide might be produced, and we can also detect it. So we need to a little bit know and understand biology. So we need some input information first place. And this is basically how we do that now. Hopefully at some point we'll be able to, that's a dream, I wish, we hope we'll be able to distinguish between different type of free radicals. So that will not gonna be any more T1, but something else, right? But yeah, so hopefully we'll be able also at some point distinguish between free radicals. And that might also then bring more answers to more fundamental questions, or how the disease develop, what are the exact free radicals involved in development of certain disease. But for now, we anyway see that something is elevated, like it's increasing the free radicals level, and that correlate with disease or not. And this is also enough. And for example, for spermatozoa, it's a superoxide mainly what we detect, if this is about the NOx5, and that's known. So then if we measure a little bit of something else like nitric oxide, I would say it's just the minor part in the signal we detect for spermatozoa. It's mainly superoxide. I haven't shown also other examples. So for example, endothelial cells, so that which are the outermost layer, so the contact layer with the blood, and those are cells are in our vessels, and those are producing superoxide and mercuric oxide as well. And yeah, we are able to distinguish, and that's more difficult there to think about it and see what's going on or how the disease develops. But yeah, maybe one day soon we'll be able to distinguish.
Thank you very much. Complex issue. Good luck with it.
Thanks. Yeah.
Mhm. I also have a question, Aldona. Do you use some targeting ligands for immobilization of nanodiamonds on membranes or mitochondria?
Yeah, that's what we do. What we did so far, we used some antibodies. I haven't shown any experiments that we use that kind of modified diamonds, but that this is what we're doing also in the group. So we modify the surface with antibodies and target to nucleus or mitochondria. So that was like one work of a colleague of mine published this year when we targeted the nanodiamonds to mitochondria, and it was like successful. Now we face the issue with the sperm cells from human source that I have to say that we will need some modification to attach the diamonds to the surface of those cells, because for Boltzmanns it worked a little bit easier. So now I know there is the technical issue to solve, but I think it's doable with the surface modification of the diamonds.
Mm-hmm. And I also wanted to ask a little bit more on nanodiamonds themselves. So on average, T1 is about 100 microseconds, right? So first question is, how wide is variation between T1s of different particles? Probably it's about twice, right? At least.
Yeah,
maybe 2-3 times. Very broad variation. Yeah,
it is exactly what, so you answer the question. So it's basically that what I also observe when I measure and analyze data. It's, sometimes it's twice. So yeah, that would be better for us to have, that the variation would be narrower, would be that big. So sometimes, so what we did also, we try to normalize values between experiments. That's also what we did. We see the initial values are different. We try to normalize it also. But yeah, that's the point. There is the difference between the diamonds.
And so you kind of just your workflow, you immobilize diamonds on, say, object of interest, and then you look, say, at few different particles, and then you pick up one with kind of longest T1, or how do you choose which diamond is?
Yeah, basically it's a kind of like we see how bright is the diamond, is how I would say it in our technical jargon, what we're talking about between each other in the lab. So, okay, this is T1, or photon counts need to be at a certain level. So we try first, we check, okay, this diamond is also, this is single diamond, or like around the smaller aggregate of the diamonds. So we don't choose the big aggregates, for example. And then ascertain the brightness range, because that works well. So we learn that's based on experience. So if it's
Okay, thank you. Please ask your questions.
I had a question. I think you mentioned that you were using oxygen-terminated fluorescent nanodiamonds and also the amino-terminated fluorescent nanodiamonds. But then you started leaning towards the oxygen-terminated ones. Why was that? What was the difference in free radical detection between these? And like, why do you think that happened?
Yeah, as I said, like there's this standard deviation. I mean, basically what we saw, like sometimes we got the, so as we average also over few measurements, then we saw like, oh, this is like high standard deviation. So somehow like NV centers are affected in this case when we use the nitrogen terminated diamonds. And I'm not sure exactly how it's that. I mean, when we took the CM images, then we see there are many nanodiamonds close to each other. Even they are not aggregating, but there are many. And I was thinking, oh, maybe there are too many close to each other. And we know that the NV centers also kind of affect each other when they are close, right? So if you have also the surface NV centers, but I don't know, maybe that's also related to the NV minus, NV zero transition. I don't know that. What's there? Maybe there are some people here who would have a bigger expertise in that area. So I cannot answer you why exactly that, but yeah, we observe that happens.
Okay, thanks.
Yeah.
Jump in with one quick question. I noticed in the beginning of your talk, you actually had some correlations from your normalized T1 values to actual micromolar values.
Yeah.
It looked like they were in the maybe like 0.6, maybe about 1 micromolar. So I was curious, like how are you doing those normalizations, and then is there a reason that you didn't keep showing those equivalencies in the arthritis work?
Yeah, I still didn't do that because it's it's still the work in progress. So this is the only reason. So I'm sorry for that. I suppose like it would be better if we just calculated before my presentation. But
so is that a consistent calculation, or I mean, are you standardizing to superoxide, or is there? I mean. Yeah, okay.
We standardized. So that's a work published already. So a colleague of mine did like more chemical work on the concentration, and is then T1 values correlated with the concentration of specific type of free radicals. So basically this is what we're using for calibration.
But so then you can actually apply that experiment to experiment.
Exactly. This is what we think. Yeah.
Great, thanks.
Yeah.
You have different types of free radicals at the same concentration, then you would have the same T1 profile, kind of dependence on concentration of free radicals. For example, superoxide versus OH radical.
Yeah, I believe yes. Right. It's like we cannot now be specific to the type of the free radicals we have in the surroundings. So I think that doesn't matter. It's just like you pulling everything and you measure this whole.
But I mean, in a controlled experiment, if you kind of on a purpose produce this type of that type, then you will get the same T1 curves.
Yeah. Basically, you can do that. So you can trigger some, for example, I can imagine you can, in the cells, usually you can switch off and switch on something. So I can imagine you can program the process, some of the processes, if you are very localized somewhere, and that you can turn off production of one radical and increase the production of different radicals. So it does need to be only for sources, but also of the type of the free radical you generate. But I'm not sure if we're doing that kind of experiments or we've done it so far. If not, perhaps that would be also interesting to do. So thanks.
I was just thinking in your response to, I think, Sean's question, this might be a pretty fundamental thing. So if it's, I don't know if it's been looked at or not, but if you were to just take a series of diamond in solution concentrations from like 1 micromole or 1 microgram per mil up to 10 micrograms per mil, would you see a difference in T1 times?
Okay, so this is what we're doing now.
So based on the idea of particles being close together, and whether and how much they actually would affect.
So this is what we're doing now. So okay, so that I don't know yet, but this is what we're doing now. So we're doing like kind of that perhaps will be soon published, hopefully. So like a lot of about reproducibility of measurements and sensitivity kind of. And also, we also immobilize on different situations like environment, the nanodiamonds, and then we expose them to different type of free radicals, and then we measure what's the T1, what's the relaxation time. But I cannot tell you now by heart because this is something ongoing. There's an ongoing experiment, and I don't know by heart the results, and some are perhaps still people or I supposed to process. So once we have it, then we will share, of course, you will see it. Yeah, so I cannot answer.
Oh, please ask more questions. Maybe then I have one more in continuation of what we are talking about. So, do you think some kind of measuring some ensemble T1, does it make sense at all to increase kind of reliability of measurements, that you don't monitor just one particles over, say, many days, but monitor, say, 5 or 10? And of course, you can have individual T1 to monitor. And then kind of combine results together to increase fidelity of measurements. Or my question is it possible to have kind of a T1 from a suspension of diamonds? Or it makes no sense?
You mean suspension? I mean, what we do?
So that you have, say few diamonds, and you get kind of this kind of your combined fluorescence from few particles, so that it would be averaged T1 over few particles.
What we do so far, maybe I mentioned it like, and this is a kind of misleading, but what I mentioned, like, okay, for the sequence of the measurements, we pick one diamond and we measure over time, but we also repeat it for different particle and different particle and different particle, and we then compare. So we average it. So we made, that's an average. So basically somehow we do that in case if the diamond, yes, in a cell somewhere on the surface of the cell. And also immobilized on the dishes different ways in different centers. But in a suspension, you mean like the floating diamonds in a suspension.
I mean just to get average T1, say, from 5 diamonds simultaneously.
Okay, simultaneously. So, but that we've never, like, the same time to measure, for example, 5 cells. That's another thing one of our colleagues is doing. But that's a little bit different. It's not with nanodiamonds, it's with diamond plates. So he's doing also kind of T1 there. But so that would be possible to do that from few NV centers, from more NV centers, and from different perhaps cells or something that cause the cell. But not with the nanodiamonds. That would be great that will like be very efficient to measure the same time few diamonds in few cells, for example, or a few locations. That would be great, but we don't do that at the moment with the nanodiamonds.
So I like to ask a follow-up question. Maybe you just said, I don't know if you said a number, but how many particles would you typically need to look at to get this average? At least 10 probably, or are you doing like?
Yeah, sometimes I see like 6 is already quite fine, consistent with the measurements, but sometimes 10. Yeah, sometimes, it's difficult because also, some chemicals we're using are, I just don't know, it's like, it's difficult to control it, especially in a biological systems, in living cells, that it's kind of a lot of things happening around, and some experiments are just not working that well. So maybe, I don't know, but answering question shortly, it's like 6-10 indeed. Sometimes we do much more. But you need to also think about it's like living cell. So we treat it with a laser. So it's still kind of effect of the laser after time. So if you want to do a lot of experiments, it's possible because we had incubator, we can provide physiological conditions, but then still you might have exposing them to the laser light. So they might don't like it that much, at least some very sensitive cells.
Yeah, I assume that the fact that it's pulsed laser is maybe helpful rather than just continuous illumination. Yeah. So
yeah, it is. It is much better. And then if you also decrease the time of the of your impulses, and then yeah, you can play with some parameters to make it more efficient and more self-friendly, of course. And this is what we also trying to optimize on a way once we're learning more. Yeah.
Evgeny Danilov, NC State. You mentioned that your setup is simpler than a quantum computer, but can you comment on the some details? What kind of laser power are you using? Is it a CW laser? What kind of pulses? How do you make those pulses?
I think this is a better question to one of my colleagues about the more technical details. So I would rather prefer, I mean, I know some numbers, but I don't want to just bring them and then say, oh no, that wasn't that. So if you're really interested, then I believe in some of our publications already, those details are there. But also, of course, I'll be happy to provide you more details later on. Yeah, we can keep this.
Just if you could give me a reference with some details of the setup.
Yeah, I don't know which paper is that exactly now. It may be already on yeasts one, but I'm not sure because, or maybe HUVEX one. So it's recently something which will be is just accepted and will be published. So I'm not sure which one, but yeah, I can send it to you if you, some info. So please,
sure.
If you could contact me, that would be great.
Yeah, absolutely. Thank you.
Yeah, thank you. And sorry, I don't know everything. This is why we're a big group.
It's great labor distribution in your group.
Yeah.
More questions? Vadim, I'm sure you have a question. You always have questions. Vadim Machalin, would you like to ask?
Thank you very much. But actually, all my questions were already asked and answered. This is why I keep quiet. Yeah, I was mainly interested in particular reasons to use one chemistry or another, but I think Aldona already commented on that. So not this time, sorry.
Well, or later, that's also really, I'll be very happy to get in a contact later or get some more questions. It's wonderful to be part of this community and yeah, exchange ideas and knowledge.
Aldona, maybe I will ask some simple question. Do you experience any problems with recharging of NV so that your, say, NV are negatively charged, then they become positive, say, neutral due to some environment? Do you kind of worry about this? Did you meet this phenomena? Recently, there are a lot of kind of publications on all this recharging of NV.
Yeah, yeah, that was something like let's say I woke up during the night one day and I said, like, oh, well, why we don't check it every time we perform measurements? And then we had the discussion with Romana, with the group. So, so at some point in the very beginning, we were checking that, but then I understand this is just so minor changes, or if that's happened for some of NV centers, it's just a part of NV center we're using because we average signal from this assemble. So So it's not that important, at least this is what we think for now, but as you brought that topic, and indeed there are some new publications on it, I still need to go through, that maybe it's good to at least control it, like from time to time you calibrate your scale, or you check your equipment. So maybe why we don't check these transitions with our NV centers and how they affect our measurements. So of course I'm open, and I think that we should do it once again. So thanks for that comment.
Another question. Do you know this silicon vacancy neutral has a spin and also ODMR property, and then also probably similar T1 behavior? Do you think it, do you know if somebody tried to test applicability of T1 relaxometry concept to other spin containing centers in diamond?
Well, I think we were trying to do that some time ago. As I understood it, I was still not a member of the group, so quite beginning. I don't know. We also discussing these things, if we want to go with different NV centers and why not. So maybe at some point we will try also different NV centers, but for now we are happy with the nitrogen vacancy centers. So those are our favorite for now, as you know. Well, so yeah, we keep working with them. But yeah, maybe at some point it's good to again go back and rethink and try again. Especially if you have some some good hints like for improvement or what might work better, then we'll be happy to talk about it.
More questions? I have another question. So how your group finds medical topics? How you identify medical problem you then start to work on?
We are very lucky because we are just located in a hospital, so that brings a lot. So we just try to keep talking to clinicians, medical doctors, but also we have many people with me included partly, right? So with a biological background, and somehow those people either work for more towards medical applications before. So somehow Romana also attracts the people who are really fascinated by the medical problems. So this is why how we get there, how we think about that. But yeah, definitely being located in a hospital is also helping or bringing opportunities. Yeah.
Because yeah, from your experience, would be great that other members of nanodiamond community can learn how to approach medical people, how to engage them, to work with us.
Yeah, our collaborators are great, and so great. Also, big thanks to them for also we doing so far. There's basically everything going smoothly. Yeah, I know it might be different. I know also from different countries that sometimes it's a bit more complicated. All the clinicians are busy. So yeah, you need to understand that somehow we need to be more flexible and more kind of try to catch them on the way. But also I see, at least in our hospital, that they are also coming to us, and they really want to talk about the things, and they really enjoy doing science. So I think we are kind of lucky, the place specific mindset of people.
Do any medical people tried to see it on your microscope and use your instrumentation, or it's just purely your group is using instrumentation?
That's a very nice question.
How scared they are still? of
Well,
Q1 relaxometer?
you know, I would say so far we don't have really medical doctor, like the clinician who's really busy with a clinical job, like during surgeries or something, that will come and sit and measure. Maybe that will happen, who knows, but we have medical students. And I'm personally working now with one very young and very enthusiastic about the science and equally enthusiastic about the medical clinical work student. She's great. So yeah, so she's learning how to work with a magnetometer, and yeah, I believe she will have soon, very soon, hands-on equipment, and will also perform some data, like treat some data. So yeah, I mean, so far it's not like maybe we provide them a little bit easier setting. I mean, to go to the operation theater with the magnetometry, that will need some more steps and thinking and how it's possible because of the optical readout. But maybe if we just give them nice tool at some point, they will use it. Yeah.
Okay, thank you very much. Last chance to ask questions. Then on behalf of everybody, I would like to thank you, Aldona, for a very nice presentation. We wish you luck.
Thank you.
Thank you for coming to the seminar series and giving your great presentation. And on December 16th, there will be a presentation of wide field microscopy, again for biological applications, which will be presented by Jean-Paul Titien from Australia. So another interesting topic, how NV centers can be used to advance our health.
Yeah, that sounds very, very nice. So I'll be there. Thank you again for invitation and all of you.
Bye-bye.
Being here. Bye.